Good afternoon and welcome to all of you. Today we have the webinar of the Women in Science that is led by Maria Mayán. But today we have Marina Garcia. Thank you, Marina, for organizing this webinar. And please introduce you our speaker today. Thank you.
Good afternoon, everyone. It's my great pleasure to introduce you, Natalia. Natalia, who I met working in the same field some few years ago. And for those that you may not know, she's now working in the CNB, starting her own lab because she's a Ramón y Cajal researcher. And he did her PhD in the same building, in the CBM Severo Ochoa. Then he did a pretty long postdoc in the Queen Mary University of London. And I guess she will be showing us today an amazing research that she did, or at least started in her postdoc, and it will be amazing, I'm sure. And I think I shouldn't speak much more because it's more important let her to explain her science. So Natalia, welcome. I'm very happy because you could join us today. Go ahead and show us your amazing science. Thank you.
Thank you. Thank you, Marina, for that introduction. So I'm just gonna share my screen, my presentation here. Okay, so as Marina just said, I very recently came back to Spain from London, where I did my postdoc, and I'm going to show you a little bit my previous research during my postdoctoral stage, and then how we are continuing this line of research now in the new lab. So I've very recently joined the Center for Molecular Biology in Madrid, the CBM, and I've started my new group. So I'm gonna talk about endothelial cell autophagy and inflammatory response. So as you all know, autophagy, it's a intracellular pathway that degrades cytoplasmic material from the cell via the lysosome. And although it was initially described as a cellular quality control mechanism, I'm just going to get out the laser here. Okay, although it was initially described as a cellular quality control and metabolic process, autophagy has been now involved in many inflammatory diseases and also metabolic diseases. So macroautophagy, or canonical autophagy, as I'm going to refer to it here today, is the variant of the autophagic process that has been most studied and is mediated by this double membrane organelle here called the autophagosome that is in charge of engulfing cytoplasmic material, protein aggregates, organelles, and then fusing with the lysosome for degradation. So the molecular pathways that control autophagy are very complex, and I'm not going to go into them today. I just want to highlight this series of proteins here, ATG12, 5, ATG1601, and WP2 that play fundamental roles in the biogenesis of the autophagus. And regarding endothelial cells and autophagy, this process has been described to regulate important endothelial cell functions such as angiogenesis, endothelial cell senescence, mitochondrial quality control, and the redox states of the endothelial cells. However, its relationship with inflammation is less studied. Now, a key component of inflammation is leukocyte trafficking across venular walls. That is very important for the efficient operation of the immune response. But when leukocyte trafficking is dysregulated, this can lead to pathological inflammation and different diseases such as atherosclerosis, cancer, neurodegeneration, obesity, diabetes, and of course, aging. So in the lab, we are particularly interested in studying how leukocytes migrate across venular walls in response to inflammation. And we've been focusing our research in studying this step here, leukocyte transendothelial migration, that occurs when leukocytes migrate across the endothelial cell monolayer. And importantly, this can happen in a paracellular manner when leukocytes breach in between endothelial cells, or in a transcellular manner when this happens across the full body of the endothelial cell. Now, this process is exquisitely regulated by the reorganization of junctional adhesion molecules within endothelial cells that are going to change and are going to traffic from the cell surface to intracellular compartments in order to be degraded when leukocytes breach the endothelial cell barrier. Now, it's very important, this process of the transendothelial migration is very dynamic, so it's very important to study it in vivo in the lab. And we do this by using intravital microscopy techniques, which allow us to follow the migration of leukocytes across the vessel walls. We do this by using the cremaster muscle microcirculation. The cremaster muscle is a very thin, highly vascularized and transparent tissue that we can put under an optical microscope and follow the recruitment of immune cells. And we can also use this platform for confocal intravital imaging, where we can label endothelial cells in red here with an anti-TCAM1 antibody that labels endothelial cell junctions, and also perform this imaging in mice that have GFP positive neutrophils. So then we can perform intravital microscopy and obtain this kind of videos that allow us to follow in very great detail the movement and the crawling of neutrophils, in this case, on top of the endothelial cells, and how they breach endothelial cell barriers. So this technique has allowed us over the years to investigate the mechanisms that regulate neutrophil and leukocyte interaction with endothelial cells. And for us in particular, it has allowed us to place endothelial cell autophagy in the journey that controls leukocyte extravasation from the blood circulation to extravascular tissues in response to inflammation. So how did we do this? The first thing we had to do was design a methodology in order to measure endothelial cell autophagy in vivo. So in order to do that, what we've done is use autophagy reporter mice that express the LC3 autophagosome marker fused to the GFP fluorescent protein. So LC3, it's a protein that is soluble in the cytoplasm, but in response to autophagic stimuli, it's lipidated and incorporated into the double membrane of the autophagosome, and therefore we can monitor autophagy by counting the GFP-LC3 positive vesicles. So this is how a cremaster muscle looks under steady-state conditions in a GFP-LC3 transgenic mouse. So we can see the muscle fibers that exhibit a lot of this green fluorescent puncti because they have very high autophagic activity, but not much else seems to be going on. However, when we subject the mice to inflammatory stimuli, such as, for example, ischemia reperfusion injury that brings in neutrophils into the tissue, we can observe how other cellular structures start to light up, particularly endothelial cells in postcapillary venules that are the sites of neutrophil extravasation. So if we look at in detail to this response, we can see that there's a transient extravasation of neutrophils into the tissue in response to ischemia reperfusion injury. And very interestingly, endothelial cells in postcapillary venules upregulate the number of GFP-LC3 positive puncti, so that means that they upregulate autophagy at the same time as neutrophil trafficking peaks into the tissue. Moreover, if we look at the localization of these puncti in the vessels of the cremaster muscle, we can also observe that most of them localize to cell-cell contacts in between endothelial cells, bringing up the question of whether endothelial cell autophagy could be regulating the interactions between neutrophils and endothelials. So then what happens in a mouse model where the core autophagy gene, ATG5, has been specifically deleted in endothelial cells? Well, we performed inflammatory assays in these mice, and we observed that there's enhanced neutrophil recruitment into the tissue of ATG5 deficient animals, specifically in endothelial cells, and moreover, inducing autophagy with a pharmacological approach diminished this enhanced extravasation, and this was dependent on the expression of ATG5 on endothelial cells. Furthermore, crossing this mouse model with a Rosa tomato reporter mouse and reconstituting the bone marrow with GFP-LC3 expressing neutrophils gave us these very nice mosaic patterns where we could compare in the same vessel segment wild-type endothelial cells and compare them with ATG5 knockout endothelial cells that are the pink ones here, and also by intravital microscopy follow the migration of GFP expressing neutrophils across these different junctions. So these experiments showed that actually most of the neutrophils prefer to migrate across ATG5 knockout junctions, both in a paracellular and in a transcellular manner, which was very interestingly. And moreover, that autophagy deficiency in the vasculature led to the establishment of hotspots, as we can see in this video, where we see a neutrophil migrating across an ATG5 knockout junction that is followed by multiple other neutrophils that migrate across the same site, therefore establishing neutrophil migration hotspots in the vasculature. So this indicated that somehow the ATG5 and autophagy deficient endothelial cell junctions should be altered, right? So we looked at them through confocal microscopy in detail, and we actually observed that autophagy deficient endothelial cell junctions, both in basal, but especially under IR, so under inflammatory conditions, exhibit these very enlarged, distorted, and aberrant structures that are enriched in junctional adhesion molecules such as beta-aderin or PCAM1. And we also saw that these proteins localized to the cell surface, and we think that these enlarged junctions are actually associated with the hotspots of enhanced neutrophil migration. So then the next question was, okay, is autophagy just affecting these two molecules, or is it a more general effect? So in order to approach this, what we did was isolate endothelial cells from these animals, from the lung microvasculature, and we assessed the levels of other junctional adhesion molecules. And we observed that not only PCAM and PCAM1 and beta-aderin, but also other junctional molecules were also upregulated in ATG5 knockout endothelial cells. However, other transmembrane proteins such as TNF receptor was not significantly upregulated, and neither did the chemokine CXCL1. We didn't see any transcriptional regulation of these molecules in knockout AT5 knockout endothelial cells. So then we wondered whether autophagy could be playing a role in inflamed and non-inflamed endothelial cells in the degradation and turnover of these junctional molecules. So taking PCAM1 as an example, we moved to an in vitro system, and we used HUVEC cells to perform antibody labeling assays. And we observed that actually PCAM1 is internalized and it traffics in LC3 positive compartments in inflamed endothelial cells. And also that ATG5 knockdown HUVECs exhibit impaired degradation of cell surface PCAM1. We performed some biotinylation, cell surface biotinylation assays, and we observed that the fraction of PCAM1 in the surface of ATG5 knockdown endothelial cells is degraded at a slower rate than in the control cells, which indicate that autophagy is somehow being involved in the degradation of endothelial cell junctional molecules in the cells. So to summarize the information that I've told you until now, this work was published a couple of years ago, and we know that autophagy is necessary for the correct transendothelial migration of leukocytes, specifically neutrophils, across the endothelial cell monolayer in response to inflammation, because when autophagy is lacking from endothelial cells, neutrophils migrate much more into the tissue, much faster, and importantly, this was associated with tissue damage responses. So the message that we have put out there in the endothelial cell biology field is that autophagy is the molecular basis for determination of neutrophil tissue recruitment in acute physiological inflammation. So now in the lab, we are very interested in exploring further roles of autophagy in endothelial cells during inflammation. And also we are interested in dissecting this little bit here. So the molecular mechanisms through which autophagy regulates the trafficking and degradation of endothelial cell junctional molecules. So I just told you that at least for PCAM1 in endothelial cells, the cell surface degradation is dependent on ATG5. This molecule traffics in LC3 positive compartments. However, doing other intracellular trafficking assays, we understood that this trafficking does not depend on other canonical autophagy machinery, such as WP2. And this was very interesting for us, and it actually brings me to tell you that autophagy processes in the cell are very diverse. So when we think about autophagy, we usually think about macroautophagy. That is what I've been talking about until now. So the double membrane autophagosome mediated process, which depends on both ATG5 and WP2, as well as other autophagy machinery, and that is usually involved in degradation. However, there are other non-canonical autophagy pathways where LC3 is lipidated to single membrane vesicles. These pathways depend also on some of the core autophagy machinery, such as ATG5, but they do not depend on other machinery, such as WP2. They're involved in multiple membrane trafficking events, so not only do they mediate degradation, but also secretion or recycling of different proteins. They've been described to play anti-inflammatory roles. And very importantly, the literature in these non-canonical autophagy pathways is increasing at a very rapid rate. There are multiple functions that are being discovered almost every month for these pathways. And they've got very broad therapeutic implications because, of course, if we are able to target non-canonical autophagy, we can selectively target certain functions without affecting the core autophagy process. So until now, these non-canonical autophagy pathways have been described in immune and epithelial cells, but nothing is known in endothelial cells. So this is, we wondered, okay, could they be playing a role in our process? So in order to answer this, what we've done is use another mouse model that I'm gonna very briefly explain, which is an ATG16L1 truncated mouse model that is sufficient in canonical autophagy, but lacks non-canonical autophagy processes. So ATG16L1, it's another molecule that is necessary for autophagy and that it targets the site of LC3 lipidation. And importantly, it has a canonical and N-terminal site that it's important for canonical autophagy because it binds to ATG5 and VP2. And it has a C-terminal WD repeat domain that it has been described to mediate non-canonical autophagy. So these mice, they have two-stop codons after glutamate 230, and they therefore lack the WD repeat domain, so they're not able to perform non-canonical autophagy. However, they retain the ability to perform canonical autophagy. So we got these mice, we subjected them to ischemia reperfusion injury, and then we looked at the LC3 positive vesicles in endothelial cells. And very surprisingly and very nicely also, what we saw is that the number of vesicles were decreased in these ATG16L1 truncated mice, indicating that non-canonical autophagy processes actually operate in inflamed endothelial cells in vivo. And moreover, these mice accumulate PCAM1 at their cell-cell junctions. And also they exhibit enhanced neutrophil extravasation responses in response to ischemia reperfusion injury, not only when ATG16L1 was truncated ubiquitously in the organism, but also in a chimeric mouse model where the bone marrow was reconstituted with wild-type hematopoietic cells, indicating that vascular and stromal non-canonical autophagy is able to regulate neutrophil trafficking in acute inflammation. So knowing all of these, our main hypothesis that we've got now in the very recently a newborn vascular inflammation and autophagy lab is that there is dysregulation of this non-canonical autophagy in endothelial cells can propagate and promote the inflammatory reaction, and that it can do so by regulating the membrane trafficking in inflamed endothelial cells, either through degradation, secretion, and recycling of different endothelial cell proteins. So in order to address this hypothesis, we've asked ourselves different questions. First of all, how is non-canonical autophagy being regulated in endothelial cells during inflammation? Does it depend on the action of different inflammatory mediators in endothelial cells or in the crosstalk between endothelial cells and other immune or vascular cells? Second, what is the protein inventory of endothelial cell autophagic vesicles? Can we compare canonical autophagosomes with non-canonical vesicles and see how different the protein inventory of these vesicles is? And finally, why is non-canonical autophagy operating in endothelial cells? In order to address this question, we aim to generate a model of endothelial cell non-canonical autophagy deficiency and subject them to different inflammatory reactions to study different functions of endothelial cells during inflammation. So just to give you a little bit of information of what we are on our latest results in the lab, we are setting up a model in order to now see whether we can detect non-canonical autophagy specifically in endothelial cells in vitro. So what we are doing is stimulating HUVECs with different activators of either canonical autophagy or such as taurine-1 or non-canonical autophagy such as monensine, and we can see that both of these drugs are able to recruit to induce lipidation of LC3, especially monensine, that it's the non-canonical autophagy inducing drug. And most importantly, the effect of taurine is abrogated upon canonical autophagy inhibitors, such as the inhibition of ALK1 and 2, and also the inhibition of the subunit of the PI3 kinase BPS34, but these inhibitors are not able to reduce the lipidation of monensine, indicating that endothelial cells seem to exhibit non-canonical autophagy in vitro, but of course, is this happening under inflammatory conditions? We've been stimulating the cells with different inflammatory cytokines, and although very preliminary, it seems IL-1 beta is able to induce the lipidation of LC3-2 in endothelial cells, and this is partially inhibited by the canonical autophagy inhibitors, but not completely, indicating that maybe IL-1 beta could be inducing non-canonical autophagy in endothelial cells. Of course, is this also happening in vivo? That's something we want to address. And what is the cellular source of IL-1 beta in the tissue? Are neutrophils particularly playing a role in and cross-talking to endothelial cells to generate some of these autophagic responses? So in order to answer this, we are also targeting this aspect in vivo. So we've done neutrophil depletion assays in the autophagy reporter mice that express GFP-LC3. So we get rid of the neutrophils, as you can see here, and we've quantified the responses of the endothelial cells that seem to exhibit a decreased autophagic response when neutrophils are not there. And moreover, in vitro, again, when we incubate endothelial cells with neutrophils, these cells, the endothelial cells, seem to upregulate the autophagic responses in contact with the neutrophils, which indicate that neutrophils actually might be instructing endothelial cells to upregulate autophagy under inflammatory conditions. Whether this is through IL-1 beta or other soluble mediators or a direct cell-cell contact, we still don't know, and something that we want to study in the future. And with this, I want to finish and acknowledge everyone that contributed to the work, especially by my very new group in the CBM, and everyone involved, my collaborators here that are helping me to set up the new lab, and of course, my postdoc supervisors and all past collaborators where I started the work. So thank you very much, and happy to take any questions.
Yes, thank you very much, Natalia. Just remember that you can do your questions in the chat, either in the Zoom chat or in the YouTube chat also. You can do your questions there. Marina, do you have any question to Natalia?
We can wait a little bit for the people, but I have some questions. Well, first of all, just saying that as I imagined before, that the work is beautiful, the science is very clean and is beautiful, and it is a little bit difficult. Came up with some question because I was writing down things, and suddenly you were answering my own thoughts. So it was like, oh my, how I am gonna ask any question now, because she's doing everything that I was thinking. So that's a tough thing. The good part is that now you have opened like a huge line of research, and I think there is some very exciting things to come, and it's very, very cool. Well, I have just a technical question or very general, and I wonder, when you did the bone marrow transplantations, have you had effects with other immune cells because you you, you transplant all the whole bone marrow and then you get difference in other like macrophages or that can affect your model. Do you have something?
Yes, so it's not an effect that it's specific for neutrophils. So I've also looked at the extravasation of other innate immune cells. So for instance, monocytes are also increased when there's no ATG5, so when there is no autophagy. In endothelial cells, the extravasation of monocytes, it's also increased. Since the only thing I have performed until now are acute inflammatory assays, we don't know whether the trafficking of adaptive immune cells could also be affected. I think it would because I think it's a very general effect. But since I've done only like the cremaster assays and also some peritonitis, the only thing I checked for was monocytes and also eosinophils. Eosinophils are also increased.
I have one question, Natalia. Thinking in diseases, have you explored or are you planning to explore if the people that is responding bad, for example, to COVID-19 or to other pro-inflammatory insults, people that develop sepsis, for example, have any of the markers of this pathway, or you can use this proteins involved in this pathway as biomarkers of the response, or have you explored any of this?
Yeah. So you mean, but regarding the autophagy responses, or you mean the junction? Yeah, so I mean, there's definitely a lot of work going on with, for example, autophagy in endothelial cells in sepsis models. It's been shown to regulate. Of course, like autophagy is doing plenty of things in the cell. So that difference in sepsis outcome may be due to the trafficking of the immune cells, but also to what the endothelial cells are secreting, or so it's difficult, I think, to point out like a specific biomarker. Maybe in the non-canonical responses we find something more specific. I don't know. That's that's what I think. That's my hypothesis about it. We'll see.
And taking into account that you have explored the connection with interleukin-1 beta signaling, have you tested inflammation inhibitors in the context of these assays or not?
No, it's something we want to do because so obviously the secretion is mediated by the inflammasome. There are also some works that describe that there are unconventional secretion pathways that maybe also mediate the secretion of this molecule. So yeah, it would be very interesting to look at that. Yeah.
And finally, are you planning to explore this pathway in the context of cancer?
Not yet. Not yet, because it's another research field, but I get that question quite a lot, because obviously the mechanisms that control leukocyte extravasation could be easily playing a role in the extravasation of tumor cells and mediate metastasis. So yeah, it's definitely something to follow up in the future.
Okay. We have a question from Nuria Casals. She says, "Good talk. I'm curious about the non-canonical autophagy. Can you explain a bit more how the cargo is engulfed? Is it selective? How do you differentiate the autophagosomes from these other organelles?"
Yeah, very good. So I mean, this area is quite an emerging research field. So how how do we differentiate the vesicles? Usually by EM unit. So you need to see whether there's a double membrane or single membrane. That's the definite characteristic of defining canonical versus non-canonical. How the cargo is engulfed, so it's not, it's a different process. It's more like a trafficking process. So endosomes, lysosomes can be suddenly coated with LC3. So LC3 coats an endosome that needs to be secreted or degraded. So it's not the formation of the whole double membrane structure. And yeah, what was the other? I think there were several questions.
Can you explain a little bit how the cargo is engulfed, and is it selective?
Is it select? Yeah. So as I said, the mechanisms are just being discovered. So, for example, there are different kinds of non-canonical autophagy. So the LC3 can be recruited, for example, to phagosomes. And that requires some machinery that is Ross dependent. Then there's other LC3 that can be recruited to endosomes. So it's always from extracellular cargo. So autophagosomes are more, usually they degrade the cytoplasmic material. Non-canonical autophagy is usually linked to things coming from the outside. Also, microorganisms are degraded by non-canonical autophagy. So yeah, I think those are the differences.
And we have another question from Iñaki that says again great work, and he has a couple of questions. The first is, does the endocytic machinery have something to do with impaired degradation of surface CD31 in the 88H5 knockout mice? In the knockout mice, does the endocytic machinery have something to do with the impaired degradation of the CD31 in the cow mice?
The endocytic machinery, is that the question? So CD31 is described to the usual endocytic machinery is involved in the degradation, in the recycling of CD35. And most of it doesn't go through the autophagy pathway. It's just that some of it does. So that's what we are trying to uncover. But yeah, the usual endocytic machinery is involved.
And the second question is, have you checked the amount of LC3 in endothelial cells from age versus young mice, both at basal conditions and under inflammation?
Very good question. We haven't because it's very challenging to work with age mice. I would love to do that because, yeah, obviously there's a clear relationship between autophagy and aging. So the aging of the vasculature is key for the development of cardiovascular diseases, and it would be great to see differences in autophagy there.
Related with aging, very great question. Yeah. So we don't have more questions from the public, but do you want to ask something more, Marina?
Yeah, I have a small question. Do you know if IL-1 beta has LIR motifs? To see.
I don't know. Marina. Yeah, very good question.
You can check easily in the website, so it might give you a clue. It might don't have, and it might be a target because sometimes they link with other things. For example, when I look for the LIR motif for PLIN3, it doesn't have. It doesn't bind with LC3 and still is a target. But it's like a clue that.
Yeah, no, definitely. Yeah, we should do that. Yeah, no, I don't know. Thank you.
You're welcome. It was very difficult to to ask you things because everything was very well.
No, but it's a very, very, very interesting talk that opens many questions for the future, and you have a lot of work to do with this new discovery. So it's just a really interesting talk. So thank you very much, Natalia, and also Marina, for organizing and for speaking and doing the talk in this meeting. And thank you all the people that has been attending also the meeting, and we will see in the next webinar. Thank you.
Okay.
Bye.
Well, thank you very much for having me. Thank you.
Thank you.
Thank you so much.
Bye. Bueno, muy bien, muy chulo.